Today topic discuss about fungal protein. Single cell protein typically refer to source of the mixed protein extracted from the pure and mixed culture of algae, yeast, fungi or bacteria used as substitute for protein rich food in a human or animal field. SCP are dried cells of microorganisms which can be dietary protein supplement. Next one, microorganisms. In microorganisms, yeast, fungi, bacteria, algae. First, yeast, Saccharomyces cerevisiae, Pichia pastoris, Candida utilis. Next one, fungi, mycoprotein, Aspergillus oryzae, Polymorphus, Trichoderma. Third, bacteria, Rhodobacter, capsulatus. Next, algae, Spirulina, chlorella. Production process. Production is carried out in the following steps. First one, selection of the suitable strain, fermentation, harvesting, post-harvest treatment, SCP process of the food. Single cell protein production process take the following steps. First, microbial screening, choice of the raw material, process of the engineering and the process of optimization, technology development, economic consideration, process of feasibility, at last safety conquer. The fungal single cell protein, some actinomyces and the filamentous fungi reported to produce the protein from the various substrate. The many filamentous fungi have been reported to produce the protein. The term mycoprotein have been introduced by the Rankovic McDowell RHM in the United Kingdom of the protein produced glucose or starch substrate. In this fungal SCP, first one, actinomyces and filamentous fungi reported in some produced the protein. Fungal production SCP. During the World War II, attempts were made to use the culture of the Fusarium and Rhizopus grown in the fermentation protein food. The inoculum of the Aspergillus oryzae and the Rhizopus species chosen because of the non-toxic nature. Sporophytic fungi grow on complex organic compound. They render into the simple form as the result of the growth. High amount of the fungal biomass is produced. The mycelial yield vary widely depending upon a microorganism and the substrate and the strain of the some species of mold. For example, Aspergillus genus niger, Aspergillus fumigatus, Fusarium graminearum are very hazardous to the human. Therefore, use of the such fungi should be avoided. Of toxicological evaluation should be done before recommendation to use the single cell protein. Next one, differentiation microorganism in fungi and substrate. Aspergillus fumigatus, the substrate maltose glucose, Aspergillus niger, Aspergillus oryzae, cephalosporin species, cellulase, hemicellulase, in the penicillium, glycopium, glucose galactose, lactose, Rhizopus species, glucose maltose, Trichoderma viridae, Trichoderma alpha, cellulose pentose, Polycellomyces variety, sulfite waste liquor, the Fusarium graminearum, starch and glucose. After SCP production by the acid tolerant fungus, the SCP production by the acid tolerant fungus, Scythladium acidopilum from the acid hydroxylate of the waste paper. The waste paper pre-treated with the 72% of the sulfuric acid and the diluted with the water and hydrolyzed. The total 97% of the sugar hydroxylate convert to the cell biomass. The based on a sugar consumed produced a high yield in check the culture. Many other fungi imperfecti. Actually, this tell about the single cell protein produced by the acid tolerant fungus with 72% sulfuric acid. After total 97% of the glucose hydroxylate converted to the cell biomass. This type of the sugar is glucose. Single cell protein production by the thermotolerant celluloid fungus. A newly isolated celluloid fungus showed 50 to 100% faster growth rate and over the 80% more final biomass protein formation than a Trichoderma viridae. The very well-known high cellulase producing the cellulolytic organisms. In both cases, Trichoderma viridae produce much higher quantities of the free cellulase at the faster rate. Also degrade most substrate and then cellulolytium. It is concluded the synthesis mechanisms are the nature of cellulase complex of two types of organisms. Different such as the cellulolytium is more optimal to the single cell production, while trichoderma is more optimal of the production of the extracellular cellulase. In this trichoderma viridae production of the extracellular cellulase. Thank you.